Okay, I'll get started here now. Just some introductions. So, welcome to SADS Foundation's webinar today, June 10th, 2025, titled Rocket Pharmaceuticals, Early Results from a New Gene Therapy Study for PKP2 ACM. I'm Marcia Baker, the SADS Foundation Program Director. This webinar is being sponsored by Rocket Pharmaceuticals. We thank Rocket for its grant support to make this webinar possible. The SADS Foundation team has been partnering with Rocket's patient advocacy team for several years now. Today, you'll hear directly from one of the lead researchers about phase 1 data outcomes and what Rocket Pharmaceuticals has learned so far from the first patients treated. Rocket Pharmaceuticals Director of Patient Advocacy, Amy Price, is also here to address your questions. So first, let me introduce you to the principal investigator, Dr. Barry Greenberg. He is professor of medicine at the University of California, San Diego, and directs UCSD's Advanced Heart Failure and Mechanical Circulatory Support Program. Dr. Greenberg is an internationally recognized cardiologist specializing in advanced heart failure, inherited cardiomyopathies, and emerging gene therapies. Dr. Greenberg is a principal investigator in the ongoing phase 1 clinical trial of RP-A601 gene therapy, bringing deep expertise in both clinical care and translational science. Dr. Greenberg has published extensively and led numerous clinical trials aimed at improving outcomes for patients with heart failure and genetic cardiomyopathies. He is committed to helping bring new hope to families affected by inherited heart conditions through patient-centered research. Joining Dr. Greenberg will be Amy Price, the Director of Patient Advocacy at Rocket Pharmaceuticals. Amy has been in the rare disease and gene therapy space for 14 years and is pleased to be supporting this PKP2 ACM community in her role at Rocket. We're honored that both Dr. Greenberg and Amy Price are joining us today, and we thank you, our community, for participating as well and hearing firsthand from the Rocket Pharmaceutical representatives. Following the presentations, you'll have time to post your questions in the chat feature. Either Dr. Greenberg or Amy will address them at the end of the presentation. A reminder that this webinar is being recorded, so please keep your microphones muted during the presentation. And the webinar recording link will be shared with all of you in a few days. So Amy, I'll now turn it over to you to share your screen.
Thank you, Marcia. Give me a moment here. While I get this ready, I loved seeing everybody's locations included. I'm based in Denver, Colorado, so I'll add mine in there as well. Thanks, Marcia, and thank you to the entire SADS Foundation team for bringing us together and facilitating this virtual space for us to provide important program updates. As Marcia shared, my name's Amy Price. I'm Director of Patient Advocacy at Rocket Pharmaceuticals. Before I turn it over to Dr. Greenberg, I wanted to share a brief overview of Rocket Pharma for those that we may be new to you. Rocket Pharma is a fully integrated late-stage biotechnology company with a vision of seeking gene therapy cures. We do this through advancing a sustainable pipeline of investigational genetic therapies designed to correct the root cause of complex and rare disorders. We are guided daily in our work by our values of curiosity, trust, generosity, and elevate. Rocket's innovative multi-platform approach allows us to design the optimal gene therapy for each condition, creating potentially transformative options that enable people living with rare diseases to experience long and full lives. In addition to our PKP2 program, Rocket's AAV vector-based cardiovascular vascular programs includes a late-stage program for Danon disease, which is a rare disease that primarily affects the heart, skeletal muscles, brain, and leads to cardiomyopathy. A preclinical program targeting BAG3-associated dilated cardiomyopathy, or DCM, which is a heart failure condition that causes enlarged ventricles. Rocket's lentiviral vector-based hematology programs consist of late-stage programs for Fanconi anemia, a genetic disease that leads to bone marrow failure and potentially cancer, leukocyte adhesion deficiency, or LAD1, which is a severe pediatric genetic disorder which causes recurrent and life-threatening infections, which frequently are fatal, and pyruvate kinase deficiency, or PKD. This is a red blood cell disorder resulting in increased red cell destruction and mild to life-threatening anemia. Across all of these programs, patients and their families are our North Star and central to the advancement of these genetic therapies. I'm now pleased to turn the webinar over to Dr. Barry Greenberg.
Thanks very much, Amy, and thank you, Marcia, for the introduction. I'm delighted and really honored to be here with this audience today. What I'd like to do over the first 20 to 30 minutes is give you a brief overview of gene therapy and some of the exciting results that we found in this early phase clinical trial, and then open up things to questions that people in the audience might have. And I think that probably that second part of the hour today is going to be the most important. So Amy, if you would advance the slides for me. As Amy is doing that, I'll give you a little bit of background about myself that's pertinent to the talk I'm giving today. I'm a cardiologist at the University of California, San Diego, and I've been involved in investigative research both on the basic and clinical level for most of my career. I've gotten interested in gene therapy probably about 15 years ago, maybe a little bit earlier because we were using it in my lab. But clinically, we started using it about 15 years ago. And I think the whole field has exploded in a positive direction over the last couple of years, based largely on some of the early work that we were able to do, particularly for a separate genetic condition called Danon disease. I'm not going to talk about Danon today, but I will talk about our early results in PKP2. But many of the lessons we learned from our Danon patients and with the treatment that we gave them have carried over to the protocol and the management of the patients in this current clinical trial. So let me start out by giving you an overview of PKP2 arrhythmogenic cardiomyopathy. This is a rare inherited condition that affects how the heart cells are connected to one another. And there is a series of proteins, which I'll describe in a little more detail in a later slide, that's responsible for holding these cells together. This complex is called the desmosome, and it's made up of a number of proteins. PKP2 is one of those proteins. When PKP2 is deficient, as it is in this condition that we're discussing today, those heart cells don't hold together so well, and that affects both mechanical effects of the heart, that is the way the heart contracts, and this becomes most apparent in the right ventricle, and also the way electrical signals are conducted throughout the heart, and this gives rise to the arrhythmias, which are so important in this condition. This is something, because it's an inherited disease, is passed down in families, and it can be seen even in young adults, sometimes even earlier. I've already mentioned it can cause a weakening of heart function and also predispose patients to developing arrhythmias. Often patients are unaware that they've had it until these symptoms are clinically manifest. Amy, let's move to the next slide. So I think most germane to the lecture today is the fact that PKP2 cardiomyopathy can lead to heart rhythm abnormalities, and this can be very dangerous. And more than 70% of patients who have an arrhythmogenic cardiomyopathy due to PKP2 deficiency experience dangerous heart rhythms. Often, when this condition is identified, an implantable cardioverter defibrillator or an ICD is implanted to help prevent sudden cardiac death. In addition, and remember, I already mentioned that the desmosome, which is now weakened, is a protein complex that holds cells together. And when that's deficient, the cells don't hold together well, and they become particularly vulnerable when they're put under a lot of stress. And one of the things that can provide that stress is strenuous activity or sports. And we've had to tell our patients that they need to cut back on these. This has been, in my experience, one of the most difficult things for the patients. And I'd be curious to hear from some of the patients in the audience about this, because many of them have been very active with sports and other uh, activities like that, and they've had to really substantially change their lifestyle because of this. There are treatments. If the heart function becomes abnormal, we can treat them with a variety of medication. We can give beta blockers and antiarrhythmic medications for the rhythm disturbances and also put an ICD in place. However, these are palliative treatments and the disease can continue to progress and get worse. There is currently no cure for this disease at this time. Next slide, please, Amy. Well, what is gene therapy? And I've already alluded to the fact that this is a genetic disorder that we're talking about. And all of us get 2 copies of the PKP2 gene. For patients with PKP2 arrhythmogenic cardiomyopathy, they get 1 normal copy of the gene and 1 abnormal copy. And this abnormal copy contains a loss of function mutation. And what that means is that the gene is not able to encode for normal protein, so that the patients end up getting only about half of the amount of the normal PKP2 protein that's so critical for the integrity of the desmosome. So they will have some PKP2 protein, but it's diminished to about 50%. The concept of gene therapy is that the aim is to supply normal copies of the gene to the cells in the heart so that these genes can set up shop and begin to produce the protein and bring that level in the hearts of these patients up to a normal range. The way we deliver these genes is to use a viral vector. This is given intravenously. It circulates around the body actually for several days to weeks, and the viruses are picked up in cells around the body, including those in the heart. And in the heart, they set up shop in the cardiac cells, in the nucleus of those cells, and begin to produce the deficient protein. Next slide. So I want to make clear, though, that gene therapy does not replace the complete set of genes in a cell or in an individual. That's a fairly common misconception that people have. And let me remind the audience that the human body comprises approximately 25,000 genes, and they're stored in the DNA and 23 paired chromosomes. Gene therapy can target individual genes, but they're only targeting one of these genes. So we're aiming to replace a copy of that mutated gene, which leads to the deficiency in protein, with a normal copy of that gene. The therapy is not integrated into the patient's germline so that offspring of the patients may still inherit their patient's genetic mutation. In fact, they'll have a 50% likelihood of doing that. Gene therapy is currently not known to directly impact fertility, but some of the chemotherapy used in some gene therapies for conditioning may increase this risk. Let's go on to the next slide. So this is a cartoon that's kind of simplistic, I think, but I maybe gets the message across. And that is that in the normal patients, the genes, the two copies of the PKP2 gene are present. This gives instruction for adequate amounts of PKP2 protein. In somebody who has a mutation in one of those genes, they only get a half dose of the PKP2 protein. So what we do by giving viral gene delivery, and here we use a virus called an adeno-associated virus, or AAV delivery system, we bring back normal copies of the gene to the cells in the heart, and the aim is to correct the underlying issue, not just to treat the symptoms. Let's go on to the next slide. So this is very exciting data, and I've just had a chance to present this at the American Society of Cellular and Gene Therapy in New Orleans in May of this year. And this is the initial results from our first phase in the PKP2 study. The primary aim, because it was a phase 1 study, was to evaluate for safety, to see if there were side effects, to determine the appropriate dosage, and also to establish the timing of when this therapy might be most effective. There were only a small number of patients. In fact, the N for this study was 3, and that's usual for these early phase clinical trials for gene therapy. In addition to looking at safety and looking at side effects and dosage and so on, we also evaluated whether or not this treatment was getting the job done. And we did that by looking at the levels of the protein in the heart and also whether or not it might have some clinical impact. And then finally, this data is going to be submitted to the FDA and other regulatory agencies around the world, and hopefully will form the basis for approval of gene therapy in this space in the future. I do want to mention that this was a phase 1 study. We will be going on to doing further evaluation. And what I'm going to present are the very preliminary results. The reason I'm presenting them and I'm so enthusiastic about this is because the results really are very, very encouraging. Before I go into those results, let me talk a little bit about eligibility for the patients and what the study process was. So we looked at enrolling patients who were 18 years or older. We made sure that they had a genetic deficiency in PKP2, because if we gave this therapy and it were for another cause, for cardiac arrhythmia disturbances, it really wouldn't be helpful. And then finally, we wanted to make sure that they were already protected from lethal ventricular arrhythmias by having an ICD implanted. The patients who we did not want to participate in the study were individuals who had other types of cardiomyopathy, some of them perhaps due to mutations in other genes, because we were targeting a very specific single gene. We were also excluding patients who had received gene therapy before due to the fact that these individuals have developed some resistance and immune response to gene therapy, and it might make the gene therapy ineffective right from the get-go. And then also patients who had severe weakness of the cardiac muscle due to this abnormality. And the reason for that is that there may have been replacement of normal cells with fibrous or fatty tissue, and that tissue is going to be less amenable to this kind of therapy. So we ended up identifying patients, and this was after a fairly extensive screening process. Individuals were then asked to come to our center in San Diego. This, by the way, was not a tough sell for those patients who were living in the Northeast or the Midwest. During the winter months, the opportunity of spending some time with us in San Diego was actually quite welcomed. In any event, when they came here, we initiated a very formal process of making sure that they fulfilled all the entry criteria for the study. And once we concluded that they were going to be good candidates, we started immune mediating drugs to tamp down the body's response to the virus that they were being given and the new genetic material. We then administered the gene therapy, and it's given intravenously, and it's a single dose. It's given over a period of about an hour and a half to 2 hours, and it's done under very close supervision in our hospital in the cardiovascular center. Patients are then kept in the hospital anywhere from 5 to 7 days, during which time they get frequent blood testing, and we see them sometimes 2 to 3 times a day. We visit with them to make sure they're feeling well, and then they're discharged. We ask them to come back and have blood drawn every day after that up to day 14. We examine them, and then after that, the blood analyses and the physical exams become a little less frequently, but patients do remain in the San Diego area for 8 to 12 weeks. Thereafter, they're discharged back to their homes, and they come and visit us every 3 months for the first year, and then every 6 months thereafter, up through a 60-month period. And what we're really interested in is making sure that the gene therapy is safe. And also, we want to get some idea about the effectiveness of this therapy. So let's go on to the next slide. So what did the study measure? The primary outcomes, again, were we wanted to make sure that this was safe, because we couldn't move forward if we were not ensuring patients and regulatory agencies that this was a safe therapy to deliver. And then our secondary outcome measures were to see if that defective protein was increased in the heart. This is critical, in my opinion, for knowing whether or not the therapy is going to be effective. We also monitored for reduction in irregular heart rhythms. All of these patients did have some evidence of irregular heart rhythms, and we wanted to see what effect the therapy might have. We also evaluated heart function through imaging and blood tests. And then we looked at quality of life. We wanted to know how patients felt. Were they feeling better? And what was the effect of the therapy on their quality of life? Let's go on to the next slide. So here are our patients, and I'll describe each of them individually. There were 3 patients, as I mentioned before, 1 male and 2 females. They ranged in age from 36 to 55 years. They had moderate symptoms, and I noticed that there are some physicians on the call here. So I'll give you the official word on this. They were New York Heart Association Functional Class 2. 2 of the patients had normal right ventricular functions. It turns out that PKP2 arrhythmogenic cardiomyopathy affects the right side of the heart predominantly, and some of these patients can develop abnormalities in right ventricular function. And we actually saw that in one of the patients who had mild to moderate reduction in right-sided function. All of the patients had taken a number of antiarrhythmic agents and beta blockers, and they're listed on this slide. I suspect many of you in the audience are familiar with these drugs. Let's go on to the next slide. So here are the effects of the therapy. The most common side effects in these first 3 participants were some mild nausea, some fatigue, loss of appetite, and some changes in lab values. These were mild to moderate in most of these cases, and they went away over time. The majority of them were seen early after treatment when the drug is first given and when the immunosuppression is most intense. I might mention the immunosuppression is given over the first month, and then the doses of the main immunotherapies are starting to be reduced after day 28, and by 3 months, all of our patients were off immunotherapy. So this is not recurrent therapy for modulating the immune response, such as we see in transplant patients and so on, where that therapy is being taken for life. All of our patients were off therapy by the end of 3 months. Next slide. So were there serious side effects? And there was one patient who experienced a group of side effects within the first 2 months of treatment. Initially, they had elevations in liver function tests, and that resolved. However, they subsequently developed inflammation of the pancreas or pancreatitis and a bacteremia, which is a significant blood infection. The patient was hospitalized for a period of 7 to 10 days, treated effectively, and all of these side effects went away. I just spoke with that patient who is now approaching the 18-month period after treatment, and he's feeling, and he has been feeling for many months now, back to normal and perhaps even a little bit better. We did not see any serious heart rhythm issues reported in any of our patients. And very importantly, we look for a rare condition called thrombotic microangiopathy, or TMA. This involves small blood clots and tiny blood vessels, and we did not see that evolve in any of our patients. Next slide. Well, what we found, and remember I mentioned that to me, probably the most critical example or evidence of efficacy that this therapy was working was to see an increase in the amount of proteins in the heart. And what's shown here are the data from one of the measurements that we made to look at protein expression, and that's a laboratory test called Western blot quantification. And what we do is we take a homogenate of the patient's cardiac tissue. We do endomyocardial biopsies to get that. and we extract the protein and we measure the amounts of specific proteins in these Western blot analyses. For patient one, this individual had very high levels of protein at baseline, and we didn't see any change over the 12 months. Patients 2 and 3 had much lower levels of baseline protein, and at 3 and 6 months, we saw substantial increases in the protein level over time. What was important is that we saw it not only at the early phase, but this persisted into a later phase. This was evidence that the therapy that we were giving actually was accomplishing what we set out to do. And to me, personally, this is the sine qua non of effectiveness of therapy. This is something that we really need to show. Let's go on to the next slide. So we also looked, remember I talked a little bit about that key protein complex that holds the cells together, that is responsible for the mechanical integrity of the heart and also for electrical conduction. When disordered, the mechanical integrity, particularly of the right ventricle, is weakened, and also the heart is more prone to developing rhythm disturbances. So PKP2 is one of the participating proteins in the desmosome. The other proteins that are there with it that we looked at were a protein called cadherin-2 and desmocollin-2. So we were curious to see what would happen when we gave this gene therapy to these other proteins. So let's go back to our patient one who did not show evidence on the Western blot, at least, of an increase in protein. We did another measurement of protein within the cardiac cells. Again, this is using endomyocardial biopsies. We stained with immunofluorescent dyes for the presence of the PKP2 protein. And what we saw in this patient was an increase at 6 and 12 months compared to baseline. What was perhaps even more exciting was the fact that this was accompanied by an increase in these other key proteins that are incorporated along with PKP2 in that critical linking structure, the desmosome. Let's go on to the next slide. This slide is actually what we look at in the under the microscope when we're looking at immunofluorescent sections and the immunofluorescent stain. Let me take you through this because I suspect many of you have not had experience doing this. At baseline are the panels shown on the left, and then we go to 3, 6, and 12 months. And we do have 12-month data in these patients. This is patient one, and what we're looking at going from top to bottom is the cadherin-2 and the plakophilin, or the PKP2 protein stains. Let's focus for a moment on plakophilin, which is right in the center of the slide. And what you see is a fairly weak to modest stain for plakophilin. Let's jump over to month 3, going from left to right, and still not much going on there. When we get to 6 months, though, it looks like there may be an increase, and by month 12, there is clearly a stronger signal than at baseline. Let's go up to the top now to cadherin, and we see a very similar pattern with increases in protein being evident at month 6 and seeing that even stronger at month 12. And then a really interesting demonstration is down on the bottom, and that's when we merge these together. What this shows is what's happening likely at the level of the desmosome. And compared to the 12-month figure where we see cadherin and plakophilin, PKP2, congregating together in the desmosome, that's a much more robust signal than we see at baseline. This is suggestive that what we're seeing is normalization of that desmosomal structure with therapy. Let's go on to the next slide. This is our patient 2, and we're looking here at the immunofluorescence. Remember, this is a patient who showed a nice increase on the Western blot. They also show enhanced immunofluorescence, and that's accompanied by increases in these other desmosomal proteins, the cadherin 2 and desmocolin 2. Let's go on to the next slide. And here's our patient 3. Very pronounced uptick in the Western blot. Interestingly, not so much for PKP2 immunofluorescence, but there were increases in cadherin and desmocolin. So that's all going in the right direction. Let's go on to the next slide. Here are some clinical parameters that we were looking at. And I mentioned already that 2 of the patients had normal right ventricular function. 1 had mild to moderate impairment in right ventricular contractility. And what we found was that that normalized at the 9-month follow-up echocardiogram. Quality of life, which we measure by a questionnaire called the Kansas City questionnaire, improved substantially in 2 of the 3 patients, and they also showed improvement in symptoms. Patient 3, really not much change in either the quality of life or symptoms. Let's move on to the next slide. And this looks at the changes in electrical activity in the heart. And let's start out by looking at the number of PVCs. Many of you have had either Holter monitors or loop recorders placed. And what we did was get a 7-day loop recorder put in place before therapy and then at various intervals after therapy. And I'm showing you the data at 12 months in patient 1, 9 months in patient 2, and 6 months in patient 3. And what we saw was a reduction in the average number of PVCs in all of these patients. In 2 of the patients, this was 60% or greater. In addition, patient 3, who had evidence of non-sustained ventricular tachycardia on the earlier baseline Holter monitor, that went from 5 episodes per 24 hours to 0. And then finally, a really interesting finding, because this abnormality is associated with repolarization changes on the electrocardiogram. That would be the ST wave changes. And what we saw was an improvement in that in patient 3, who had the big drop in PVCs and the reduction to in NSVT. So some electrical phenomena that are going in the same direction as the protein and histologic phenomena that I showed you earlier. Let's go on to the next slide. And so what have we learned so far? And we believe that these results that I'm showing and sharing with you today are very promising in patients with this PKP2 arrhythmogenic cardiomyopathy. We found the treatment was well tolerated. There were no lasting serious side effects, and only 1 of these patients had a side effect that resolved fully within 2 months. We did show that we were able to accomplish what we set out, and that is we got protein into the heart, and there were early signs of better cardiac function, and this led to strengthening of the heart by increasing these key proteins. These changes were most notable after 6 months, and they may well continue to grow over time. We're currently evaluating that. So I'm going to stop at this point and really thank you again for the opportunity of sharing this for you. I'm going to be able to take questions for about 10 minutes with you, and then I've got to jump off for another meeting. But again, I really appreciate this opportunity.
Marcia, do you want to do questions now if any have come in ahead of Dr. Greenberg needing to hop off?
All right, let's do that. So this is your opportunity for our Q&A. If you'll type in your questions into the chat feature, we'll get to as many as possible here. Thank you, Dr. Greenberg, and thank you, Amy, for your support and presentation for this webinar. Thank you. All right, there we go. So Danny asks, are these findings and mechanisms also transferable for people with ACM that have the DSP gene mutation?
Boy, that's a really good question, and they are not. This is specific for PKP2. We would hope that other therapies, gene therapy and other genetic therapies, could be developed for the DSP. But right now, that's not being considered because of the size and complexity of that gene.
Okay, thank you. Dr. Zariba says, congratulations. Were these patients on antiarrhythmic medications during follow-up?
So 2 of the 3 patients were. That was not changed during the follow-up period. They all remained on the same regimens.
Okay. And a question from Dr. Gasparetti. Questions regarding the pancreatitis episode. Any idea what triggered it? What kind of management was required?
So we're not quite sure what triggered that. I believe the patient probably had an episode of cholecystitis. He had some right upper quadrant pain and then developed pancreatitis. He was treated in the hospital with really conservative measurements. He developed bacteremia. We treated him with antibiotics, fluid, kept him NPO, and fortunately, this all resolved over a period of about a week to 10 days. So nothing dramatic was needed in this case.
Would you consider giving to genotype positive but phenotype negative patients?
I think that while we're testing this, since we're looking at efficacy signals, unless patients have some evidence of the arrhythmogenesis, that is, they have PVCs or they have episodes of ventricular tachycardia, they would not be treated. When this be, if and when and if, hopefully, this becomes available, that's going to be an interesting question about treating patients prior to the demonstration that there's a clinical phenotype, because we know that these can present over time, and also the first presentation can be serious ventricular arrhythmias or even sudden cardiac death. But currently, for the clinical trials, we do need a clinical phenotype for enrollment.
Okay, thank you. Did all 3 patients have different pathogenic PKP2 variants?
Oh, great question. I don't recall. I have to go back and look at that.
Okay. When will phase 2 start, and will you have facilities in Europe?
So yeah, we do want to get started on phase 2, and I'm going to have to ask Rocket to answer that question since I'm at the University of California in San Diego, and they're going to be setting up different centers.
So our goal is to start a phase 2 study sometime early next year. So that is our plan. But once we have more information, we'll make that available. And this is intended to be a global study with US, Europe, and UK to be the clinical sites. So I did see the question about UK. We are considering that. Please read for more information. Right now, we're digesting this data, working with Dr. Greenberg and understanding the patient endpoints that are valuable to the patient and what's considered benefit risk. So we are talking through that internally and with our experts, and then with health authorities around the globe, including the FDA.
Okay, thank you. Is there any attempt to give the gene therapy without 3 months of immune suppression? And can the AAV vector act and deliver its therapy before the immune system develops a targeted response?
So, boy, that's a really good question. We try to give what is the minimal effective immunotherapy, but some degree of immunotherapy is required for these patients. As I mentioned before, the immunotherapy is completed in general by 12 weeks after the initiation of this. And one other point, then it's escaping me now that I wanted to make in response to that. The answer is that you can't get enough of the therapy to work before the immune system kicks in. And even if you had delayed the immune therapy for a period of time, there's still circulating virus that goes on for a period of weeks, and that is going to activate the immune system. So currently, for the therapies that were given with the AAV virus, which is the one that we found is most conducive to going to heart tissue, we are requiring immune therapy.
Okay, thank you. Dr. Pitt asks, do you anticipate that your immunosuppressive regimen will allow repeat dosing?
So the answer is the immunosuppressive regimen will not allow that. However, there are a number of laboratories around the country that are working at ways of reducing that acquired immune response that people retain for a sustained period of time. These laboratories are working at finding ways of modulating that to the extent that additional gene therapy could be given in the future. Right now, this is not ready for use clinically. The hope is that developing some of these approaches will allow that in the future. But at present, we tell patients that at this time, future gene therapy would not be possible.
Okay, thank you. Should the level of PKP2 protein be considered to qualify for therapy? From Dr. Zarebra.
No, I think that what we saw from our patient one, they had high levels at baseline. It's hard to know what that means, whether or not, since we're measuring that protein level in the context of other proteins in the heart, whether or not all of the cardiac proteins are upregulated in these patients or not. So that the absolute level that we get is not going to be a criteria for moving forward. What we know is that when the gene mutation, and these are loss of function mutations, are present, these patients don't have enough protein in the heart for adequate function.
Okay, thank you. Theoretically, is the treatment meant to be applied before the disease has developed or manifested, or young gene screened patients?
So right now, it's really an extension of the phenotype question. We require for evaluation in this clinical trial for the patients to have the disease clinically manifest, because we are looking for evidence of efficacy. I can envision, if this turns out to be effective, that in the future you would treat patients earlier in the disease so it never becomes clinically manifest.
Okay, thank you. What immunosuppression drugs and dose are being used in this study?
So there's the foundation. There are 3 drugs. One is a monoclonal antibody called rituximab or Rituxan. That's given as pre-treatment prior to the administration of gene therapy, and that really tamps down the immune response. Around the time that therapy is given, we also initiate steroids and a drug that we use in our transplant patients called sirolimus. The dose of prednisone is really dictated by the patient's body weight up to a maximum of 60 milligrams per day. Beginning at about day 28, we start down titrating that, and by the end of 12 weeks, both the prednisone, the steroid, and the sirolimus are taken off completely.
Okay, thank you. Couple more questions is all we have time for. Here's one. How much do you think SE were related to immunosuppression versus the actual gene therapy product? Wasn't sure if you mentioned the timing of onset.
So for the side effects, some of them are due to the gene therapy, no question about that. But some of them are also manifestations of the immunosuppression. For instance, our patient developed a serious infection. He may have been more prone to that because of the immunosuppression that he's gotten. That's the reason why these patients are so intensively followed up, and also why we've modulated the gene therapy. I mentioned earlier on that I've been involved in this area for some time, and I've worked with gene therapy programs for other conditions. We've really modified the gene therapy immunosuppression regimen to try to use the minimal dose that's going to be effective.
And would you get better uptake by giving angiographic rather than IV? Presumably much of virus taken up by non-cardiac tissue.
Yeah, no, there really, the majority of clinical evidence suggests that there's not much advantage of giving it directly intracoronary with a coronary angiogram or something like that.
Okay. Can you elaborate on a timeline in years on how long phase 2 and FDA approval would likely take?
Uh, I'm going to pull out the crystal ball here, and I would imagine 2 to 3 years. But I don't really know. They could be fast tracked. Well, it really depends on how quickly the phase 2 goes, and getting this. The FDA appropriately spends a lot of time going over this data, and they should, because it's an important decision that they make. But I would think somewhere around 2 to 3 years.
Thank you. I know you need to run, so thank you very much for your presentation, and we thank Rocket Pharmaceuticals for its support in making this possible. So thank you, everyone, and have a good rest of your day.